Isn't it crazy? Our bodies are made up of thousands of genes, and these genes have determined everything about the bodies we have today. But in the creation of an organism, the genetic process can get a little murky. An example of this is chromosomal aberrations. A chromosomal aberration is a change in the physical structure of a chromosome. So let's look at an example. In the synapses of chromosomes in individual 1, we want to determine if there is a chromosomal aberration, what will result from a crossover, and if viable gametes can be produced from the chromatids. The chromosomes of individual 1 are GHI, centromere JKLM, which we'll call chromosome 1, and MLI, centromere JKHG, or chromosome 2. Now the first thing we need to do is decide if there's an aberration, so we can compare the chromosomes, see which loci are identical and which aren't. From this we see that I, centromere JK, are of the same orientation in both chromosomes, but the remaining loci are different. We can also see that HGML in chromosome 2 is an inversion of LMGH in chromosome 1, making chromosome 1 normal. Because the inverted region does not contain a centromere, the aberration is a paracentric inversion. Now in performing synapsis, we want to produce the most matching pairs possible, but since there is an inversion, we need a compensation loop to do this. We know the first two chromatids from the synapsis will be GHI, centromere JKLM, and GHKJ, centromere ILM. Let's call them chromatids 1 and 2. But let's remember there's a crossover of genetic info between the J and K loci. So we follow chromatids 1 and 2, so we're going to find the third one. If we start here, we'll have GHI centromere J crossover KHG. So our third chromatid is GHI centromere JKHG. And as you see here, there's 4 chromatids, so we have one more. But since these paths have already been completed, we're going to start on the right side. So for this last chromatid, it's going to be MLK crossover J centromere ILN. Now that we've determined the aberration and crossover results, we need to see if there will be any viable gametes. We know the chromatid can have viable gametes if it has all loci G through M, and if there are no duplications or deletions, meaning every locus is present once. In chromatids 1 and 2, we see that they are both viable, but chromatid 3 is missing L and M and repeats H and G. Chromatid 4 is missing H and G and repeats L and M. So 1 and 2 can produce viable gametes, but 3 and 4 cannot. Let's look at individual 2. The chromosomes of individual 2 are MLI, centromere JKHG, chromosome 1, and GHKJ, centromere ILM, chromosome 2. Now this one is interesting because when the centromeres are aligned, the chromosomes are identical, but if we compare them to individual 1, we'll see they are identical to chromosome 2, making individual 2 a paracentric inverted homozygote. Once we draw the chromosomes, we can follow the chromatids. We know chromatids 1 and 2 are the same, so let's consider the crossover event between J and K. If we start here, the chromatid will be MLI centromere J crossover KHG. And for the fourth, we'll start here, making the chromatid MLI centromere J crossover KHG. And since all the loci appear just once, we know they can all produce viable gametes. Since individual 1 is heterozygous and individual 2 is inverted homozygous, the fitness for individual 2 will be relatively higher. Heterozygotes will not produce as many offspring and typically don't survive as often, reiterating the lower fitness of individual 1. I hope you enjoyed this video. Here are my sources, and thank you so much for watching.